So we can move on to the next item on our agenda, agenda item 11. And this is a presentation by Dr. Brian Lapointe from Harbor Branch and Florida Atlantic University. The study is called Groundwater and Surface Water Contamination by Septic Systems in the Charlotte Harbor NEP Watersheds. And the purpose of this presentation is to provide a briefing on the latest research related to septic tank pollution and how the condition of the septic tank system and where it is placed can contribute to nutrient pollution into ground and surface waters, leading to water quality and ecological health declines. This relates to water quality improvement priority action objectives 4, to reduce wastewater pollution, and 4.2, supporting conversion of septic to sewer. Dr. Lapointe, do you want to take it from here?
Sure. Thank you, James.
Thank you.
So, yeah, what I'd like to talk today about is some of the research I've been involved with in the Charlotte Harbor NEP watersheds, really going back to about 2004 or so. Next slide. Yeah, I know some of you know me, but I think many of you may not know me. So a little bit of my background. I began working with nutrients and algal blooms at Harbor Branch. That's a photo of me in 1974 with that flowing experimental seawater system where we were dosing fast-growing red drift algae like Gracilaria with mixtures of sewage and seawater from the Indian River Lagoon. And you know, the algae grew really, really well on that sewage seawater mixture. And of course, as we'll see in my talk, we don't have to grow these anymore in these experimental systems. We have these large-scale red drift algal blooms and other types of drift macroalgae as well. This was all happening, actually. I did a lot of this work for my PhD at USF with Clinton Dawes at the time when Gracilaria and Ulver were forming big blooms in Hillsborough Bay, and that was a big part of the impetus to begin nitrogen removal in Tampa Bay. A few years later, of course, harmful algal blooms expanded nationally, and I was a group of scientists that convened at Snow Mountain Ranch, Colorado, to help develop a national research agenda called the EcoHab program, which is still functioning and run through Don Anderson's lab at Woods Hole. And I was brought in with Ivan Valiella specifically to look at macroalgae. And at that time, I was studying macroalgal blooms in the Florida Keys and Southeast Florida, in many cases in areas around septic systems here in the Florida Keys. And then more recently, actually 20 years ago or more now, I helped contribute to the book Clean Coastal Waters, published by the National Research Council, on really the fundamental problem here is the accumulation of nitrogen and phosphorus in fresh water on its way to the sea. So next slide. So I think we're all very familiar with some of the critical issues we're facing in Southwest Florida on nutrient, microbial, and contaminant pollution. Of course, the nutrients are leading to harmful algal blooms. We've had these for many years, red tides, red drift algae, and most recently blue-green algae. Loss of seagrass, decline of fisheries, emerging diseases and health impacts in wildlife and humans. And Charlotte Harbor, of course, is impaired for nutrients, dissolved oxygen, chlorophyll A, shellfish, bacteria, and fish tissue mercury in different segments of that system. You can see the map on the right is the fecal coliform 10-year average compiled in June 2015, just showing, you know, the high values greater than 400 CFU in the Port Charlotte area, an area where we have a lot of septic systems. And of course, it's not just fecal coliform, but as I'll be showing, nitrogen also is coming from septic systems into that part of the system. We see another hot spot there near Hancock Creek in North Fort Myers area that I'll be talking about as well. Next slide. So the problem of septic systems obviously has grown with population growth in the Charlotte Harbor watersheds. We have two plots of population growth. The left is Charlotte County, the right Lee County. And we got to work with Charlotte County staff several years ago. And when we put these historic data together, you see not just the population curve in blue, but also chlorophyll A, the black dots, and how they have gone up with population growth. And that red line is the numeric nutrient criteria for the system. You can see we're well above that. And of course, the concern here is that these increased chlorophyll A values that are phytoplankton in the water are reducing light needed by seagrasses. And I know most of you are familiar with this very serious problems, the catastrophic loss of seagrass in the Indian River Lagoon, where through elevated chlorophyll A from brown tides, cyanobacterial blooms, and macroalgal blooms, the reduction in light has led to greater than 95% loss of seagrasses, which unfortunately has been a major contributor to manatee mortality through starvation in that system. For Lee County, we see the population growth curve. We haven't put the historic chlorophyll A data in there yet, but that's something we hope to work on in the future. Next slide. So Florida has a lot of septic systems. So, about a third of households rely on septic systems. I think we have nationally about 12% of all the septic systems in the United States are here in Florida. And of course, we have very sensitive water bodies, springs, estuaries, coral reefs that are very sensitive to contaminants and nutrients in particular. Around Florida, for the most part, the soils are not suitable for septic systems. They don't work so well because of the porous sands and karst limestone. You have very low organic carbon content, which is required for things like denitrification, bacterial activity that can remove some of that nitrogen. And a big problem also are the high water tables. And with sea level rise, this problem is getting worse. I'm going to talk a little bit more about that in a minute. The effluent contaminants, of course, are all those associated with sewage, nutrients, pharmaceuticals, hormones, bacteria, viruses, metals. And a modeling study by Bedruzman and his co-authors in 2012. This is a modeling study that basically concluded fertilizers were the largest nitrogen source in the state of Florida, but they were followed by septic systems. Now, that's a statewide model. And as I'm going to show today, in the systems I'm working in the Indian River Lagoon and the Lee County area, Charlotte County area, the data we're getting from measurement, not from modeling, from measurements in the water are actually showing wastewater and largely septic systems are, we believe, in these urbanized areas, the leading source of nitrogen driving these harmful algal blooms in these systems. You see atmospheric nitrogen, reclaimed water are also other sources of nitrogen that are also contributing to the blooms we're seeing. So that little diagram on the right, the drainage field there, I want to talk just a little bit about the problem of the high water tables. Next slide. So these plots are from the work we did both in Charlotte County and more recently in Lee County. So the state code calls for 2 feet of separation between the drain field and the seasonally high water table in the wet season. Okay, that water table goes up and down through the year as a function of wet-dry cycles. So when you add another 1.5 feet that is generally required for the installation of a septic system from the grade level of the soil, you really need about 3.5 feet minimum separation, okay, between the top of the soil and the seasonally high water table. So in Charlotte County, based on a lot of really great monitoring well data that were available to us, 71% of those values we measured in Port Charlotte areas using septic systems, 71% had less than 3 feet. And in North Fort Myers, Hancock Creek, Powell Creek area, 87% were less than 3 feet. And in fact, in that Lee County study, you can see the wells that actually had greater than 3.5 feet were mostly the reference wells that were outside of the project area. They were on higher elevations. So clearly we have a lot of septic systems in very low-lying areas where they just don't have enough separation to function the way that they could with a greater vadose zone or aerobic zone between the drain field and the water table. Next slide. This is just a summary chart just showing some of the results from the North Fort Myers study. And this is for groundwater. We have a reference site at the top. Hancock Creek, Central Drainage Feature, and Powell Creek are the three kind of sub areas that we sampled for bacteria, pharmaceuticals, the artificial sweetener sucralose, and dissolved reactive nutrients and stable nitrogen isotopes. And we see we did find in some cases high bacteria present and high BOD in the groundwater. In fact, some that exceed state standards. Pharmaceuticals, we found some kind of, medium to low values. Artificial sweetener, sucralose, we're finding that to be a really excellent tracer of human waste. It goes right through sewage treatment plants and it goes right through septic tanks. And it's widely used. And you can see some very, very high values exceeding standards. And for nutrients, we see that we have very high values for nitrogen, particularly ammonium. And look how ammonium is medium to high compared to nitrate, where we see lower values. That goes back to the point that we don't have enough separation in those drain field systems to get nitrification from ammonium to nitrate. And without that step, there's no way you're going to get denitrification and removal of that ammonium. And of course, ammonia being the most reduced form of nitrogen, that is that is the best form of nitrogen to support an algal bloom like these blue-green algae blooms that we're seeing, or red drift algae, or red tides. And we see phosphorus, soluble reactive phosphorus, SRP, total nitrogen, total phosphorus, all very high and exceeding standards, and N15 values over here. Interestingly, the reference well was relatively low, as was one station in Powell Creek, but you see very high values. This is aqueous N15 in the water traveling through the groundwater, okay? So next slide, we're going to look at the surface water in these same study areas, where now we see very high values of enterococci above standards. And pharmaceuticals, for the most part, we'd find a few sites with medium values. Sucralose is diluted down more in the surface water, kind of medium values, but present. And here now we see the nitrogen nutrients, ammonium nitrate and dissolved inorganic nitrogen, at medium values, not really high values, because these surface waters where you have algae, these algae are assimilating those nitrogenous nutrients because it's a very nitrogen limited system. And you have residual phosphorus in the water, the high soluble reactive phosphorus in red and the high total phosphorus. So this is what you would expect to see, a residual buildup of the next limiting nutrient. So what's happened here is that wastewater nitrogen has gone from being in the dissolved reactive form in the groundwater into the particulate form. Those N15 values are measured in particulate organic matter in the water, algae, right? So basically what's happened is it's gone from the groundwater into the surface water and being taken up by the algae at high levels, high values greater than 3 parts per mil, which is in the wastewater nitrogen range. Values below that would fall into the synthetic nitrogen fertilizer range. So we are interpreting this to be wastewater nitrogen being taken up by the algae in the water. Next slide, please. This is just some of the historic work we did with Lee County back in 2004, where we came to basically the same conclusion that wastewater was a primary source driving these blooms of red drift macroalgae that began around 2003. The N15 values in that study ranged from 5.7 to 7.1 parts per mil. Again, values below 3 would fall into the range of synthetic fertilizers. Next slide. Red tide, 2005, we had a lot of rainfall from a very active hurricane season in both 04 and 05. That's my colleague Charlie Yench collecting red tide off Sanibel in September of 2005. The values of the nitrogen isotopes ranged from 6.8 to 9.5. Again, solidly in the wastewater nitrogen range. And when you look at the red tide distribution of the state database that was compiled by Larry Brand and Ann Compton and published in the journal Harmful Algae, even though these blooms are seeded by Karenia brevis coming from offshore, that's what's generally thought. You can see the very high biomass blooms, greater than a million cells per liter, are very close to shore within that near shore area, influenced by drainage from land and this wastewater nitrogen. Next slide. Of course, we saw a repeat of that 2005 bloom in 2017, 2018, with another major epic runoff event from Hurricane Irma. Came ashore at Cudjoe Key and went up the southwest Florida coast and really drenched, a real gully washer throughout much of Florida. And of course, here are the national headlines bringing back the point I'm trying to make, that we have a real wastewater infrastructure system in Florida that becomes all too obvious when we have these stress tests with climate change driving more frequent and heavier rain events. And of course, this brought back the point that the American Society of Civil Engineers made a year before Irma in 2016, giving Florida's wastewater infrastructure an average value of C, and estimated that we need to spend on the upwards of about 18 billion to bring our infrastructure up to standards. This is the point I made to Senator Debbie Mayfield and her committee back on January 9th, 2019, that helped moved that discussion along and ended with, of course, the Clean Waterways bill that passed, was signed by the governor, which is going to help local governments do things like septic to sewer and wastewater upgrades. Next slide. Just wanted to show this last data set, actually two more slides of a little bit of data, again, to show how these estuaries, both the St. Lucie Estuary and the Caloosahatchee Estuary, are enriched with wastewater nitrogen. Here's the stable nitrogen isotope values on the left in microcystis, in the blue-green algae, in the 2018 bloom, going from Lake Okeechobee down the Caloosahatchee at the locks into the North Fort Myers and Cape Coral area. Look at how those values go up, going from the lake into the estuary, okay? That is what we're seeing, is the increasing influence of sewage nitrogen going downstream into those highly populated areas. And you see the dissolved inorganic nitrogen data, again, coming across. We certainly have high values in the Kissimmee River and the lake, but look how they go down at the Franklin Lock in North Fort Myers, again, where we had that bloom in place, removing the limiting nutrient, nitrogen, down to low levels, whereas the phosphate down on the bottom graph, you can see how there was still residual phosphorus in that water, as it wasn't the key limiting nutrient. But of course, the main point of this is, look at Cape Coral. That's the Normandy Court Canal that was supporting that big bloom in the background of these data. Those really heavy blooms are clearly being supported by very high concentrations of nitrogen and phosphorus in the water. And next slide. And this is the last data slide. This gives an even more synoptic or larger scale view of these reactive nitrogens species and phosphorus and N15. Look at ammonia going from the St. Lucie Estuary, Station 1, all the way up the C44 system, across and around Lake Okeechobee, and then down the Caloosahatchee River Estuary. Okay, that's ammonium. You see both samplings, okay, October 2019, July 2020, very high values in both these urbanized estuaries. You come down to phosphorus, you see the same thing, relatively low values in the lake compared to these urbanized estuaries. And then the N15 value, this is of the particulate organic matter itself. You see that value of 3, where the values are generally well above that, where you're having the major wastewater inputs in the estuaries, compared to Lake O, which is lower, indicative more of rainfall or fertilizers that have those lower isotope values. So this clearly shows that wastewater nitrogen, once these blue-green algae blooms are in those estuaries, they are growing in place on the nutrients from that surrounding basin. Next slide, please. Yeah, and this is going to be a wrap here. I just want to thank a lot of people, the Charlotte County Board of County Commissioners, and many who helped with our Charlotte County study, and also Lee County, the Board of County Commissioners, Lisa Krieger, Roland Ottolini, who I've been working with most recently. Others, the Lee County Environmental Lab, DEP, and of course my own team at Florida Atlantic University, Harbor Branch, Rachel Bruton, Diana Bellotti, Kevin Tyre, and Brad Bedford. And I think that's it. One last slide, I think. And that's a picture of, sampling Lake Okeechobee back in 2018. All right, and that's it. Thank you very much for your attention.
Thank you, Dr. Lapointe. Great presentation.
Yeah, these are a couple extra slides I just threw in. Um, I was trained early on at Woods Hole Oceanographic. These are some red drift algae blooms that are happening right now on the south shore of Cape Cod. It's an invasive red alga, but it's being fed largely by septic tank effluent. Some of my colleagues up there, Brian Howes, have done modeling studies of nitrogen coming into these coastal waters, and they um, they concluded 75 to 80% of the nitrogen loading in the bays along this coastline are coming from septic systems. And so, of course, that looks pretty familiar to what we just saw in Lee County. And then one last slide, I think, James. So this is the solution, and I'm very proud to say, here in the Florida Keys, I think we have an example of what we need to do in other areas of Florida, like Charlotte Harbor. This is the Cudjoe Regional Advanced Wastewater Treatment Plant. Um, the influent nitrogen is about 80 milligrams per liter. There's about 1 milligram per liter coming out of the plant. It's removing 98% or more of the nitrogen. The technology now is so good. The Barden Foe 5-stage system is just amazing. The removal efficiencies that they're seeing these days. And I will also point out that this plant, shortly after it went online, was hit by Hurricane Irma. This is where Hurricane Irma came ashore, and there were no sewage spills in the Florida Keys, even with a major hurricane event like that happening.
Excellent. Thank you, Dr. Lapointe. Appreciate the presentation. Great work that you're doing. I'm going to go ahead and open it up to questions for anybody that has a question. Either raise your hand or go ahead and ask Dr. Lapointe your question.
James, this is Roland.
Roland, go ahead.
Good morning. Yes, it's been great working with Dr. Lapointe and his folks at the Harbor Branch over the years. This information is really invaluable to us for get an assessment of where we're at with our septic issues in Lee County. And we've gone beyond this and also moved forward to looking at a county-wide effort as well, because we have over 70,000 septic tanks attributed to Lee County in unincorporated Lee County. So, and as you probably realize, that our BMAP, Caloosahatchee River BMAP, now is taking a focus on septic inputs to nutrient loading to the river, and so this information can be very valuable to us in moving forward, because part of that BMAP is to develop a wastewater plan for a county and looking at septic to sewer or alternative conversions over a course of the next 10, 20 years. So again, many thanks to Dr. Lapointe and his folks.
Thanks, Roland.
Yeah, thanks, Roland. Great comments. Any other questions or comments? We have a good bit of time, so if you have any questions, go ahead and feel free to ask Dr. Lapointe.
James, this is Lisa. Can you hear me?
I can, Lisa. Go ahead.
But anyway, I also wanted to mimic or repeat what Roland said. It's been a real pleasure to work with Harbor Branch. Brian and his team are great to work with. They've done an outstanding job. We're working on another big project with them, and we really appreciate the lucidity, so to speak, that they've brought to us on this, surface waters in Lee County. So thanks, Brian. It's good to see you.
Oh, thank you, Lisa. Listen, we've enjoyed it as well.
Any other questions?
If we don't have any from our members, I have a couple.
Yeah, and I have a couple as well. Go ahead, Jennifer.
Well, I'll just start with one and take turns and see if other people have questions. But
Sure.
You mentioned the separation to groundwater as being a key feature of the 3-foot separation. What was the distance to surface waters that you found to be adequate? Obviously the proximity to surface waters is also a factor. Do you have a distance that you felt was kind of the average adequate separation on that end?
That really wasn't part of the study. Most of these homes we studied were in close proximity. They were directly on canals. Okay, but That's something we could perhaps in the future look at that and come up with an answer to your question, Jennifer. My gut feeling is that it's going to be quite a distance, in Florida. I know other areas of Chesapeake Bay and Cape Cod. They start first with septic to sewer at the worst case scenarios where these septic systems are in close proximity to the water and work back from there. But given the groundwater flow rate, even old homes, quite a distance, from surface waters, eventually those plumes over time, when we're talking decades, can eventually reach surface waters. And I think, unfortunately, that's what we're seeing up and down places like the Indian River Lagoon and in Lee County as well.
So, Dr. Wood, I have a follow-up question. It's a little different, but when you were looking at the stable isotope analysis for your fertilizer component, did that include livestock runoff as well as fertilizer?
No. Livestock would have a more enriched value of N15. I'm talking about synthetic fertilizers, say that commercial applications would apply, to landscape, urban landscape.
Okay.
But again, if you look at Lake Okeechobee, you saw how low those values were in Lake Okeechobee. And I think that mirrors a recent study that also found that the livestock north of the lake were not major contributors
Okay.
to nitrogen compared to fertilizers.
Compared to
Okay.
fertilizer, right? Okay, that's helpful. And then as we look at the the loads that come into the Caloosahatchee watershed, based on some data that was presented by the Water Management District in water year 2014 to 2018, about 12% of the load from phosphorus came from the tidal basin, and about 14% of the total nitrogen contribution came from the tidal basin. But 40% of the nitrogen came from Lake Okeechobee, and about 46% came from the C43 basin east of S79, as well as the S4 basin. So, a large portion of the total load is coming from east of S79. Now, what contribution do you think that has on, on algae within our watershed and our coastal waters?
Thanks for asking that question, James. We talked about this very issue recently in the Sarasota Bay macroalgae workshop. I don't know if you attended that, James, or
I
not.
wasn't able to attend that section.
I know Jennifer did. So there's a disconnect, I think, with these algae blooms and the modeling and the estimates for nitrogen contributions from Lake Okeechobee versus the basins. And we published this point in a paper in the St. Lucie Estuary, a study we did of septic systems, because a lot of people refer to total nitrogen, right? When they make these estimates. The total nitrogen measurement is measuring everything in the water, detritus, anything in that water, the particulate matter, in addition to ammonium and nitrate. It's only the ammonium and nitrate in the phosphate, the reactive forms, that are driving these blooms. So if you want to know what's really driving these blooms, you really have to look at those forms. So the NNC that we have adopted in Florida are all based on TN, TP, and chlorophyll A. So they don't really do a good job in terms of protecting our waters from ammonium, nitrate, and phosphate, the dissolved reactive forms, as it pertains, for example, to macroalgae blooms. So if you look, I suspect that in these estimates, they don't look at the pools of ammonium and nitrate and phosphate coming from the lake versus the basin, okay? They're doing more of a broad brush, total nitrogen, total phosphorus, okay? So that can be a bit misleading when it comes to the drivers of these blooms that we're seeing. And that's why my work, which is focused really on algae growth and and what's driving it, what forms of nitrogen, nitrate or ammonium or phosphate. I don't look at total nitrogen, all right? And none of us do that really study the algal bloom dynamics.
Okay, excellent. Well, thank you for that. I appreciate that. And then the final question I had was, as we look back on 2018, the cyanobacteria that was coming into the Caloosahatchee, I mean, we know that it was also moving in from the lake because we had an extensive bloom. And right now there's, as of 2 days ago, there was a 500 square mile bloom on Lake Okeechobee. So we are starting to see some of that cyanobacteria moving into the Caloosahatchee. In 2018, a number of people were hypothesizing that as the cyanobacteria breaks down, that it's releasing biologically available nitrogen that was taken up by red tide as an available source of nutrients to help fuel or prolong or exacerbate the red tide bloom in the coastal waters. What are your thoughts about that issue?
Well, yeah, I do think that's a plausible mechanism. In part, it can't explain it all, but when you have very high biomass blooms, whether it's red tide, blue-green algae, or red drift algae, macroalgae, when they die and decompose, those nutrients can be cycled. And in fact, and James, you've seen this. In 2004, we had the macroalgae bloom, right? 03 and 04, the red drift algae blooms, at a time when the water was relatively clear and you had enough light to support a macroalgae bloom. Then we had all the runoff, okay, from the estuary, from the basin, and some from the lake that basically set up a condition for that red tide in 05. A very prolonged red tide that blocked the light, and it basically resulted in die-off of the macroalgae. So that nitrogen and phosphorus from the macroalgae was additive, right, to the nutrients available for the red tide. And you could see how the isotope values were very similar between the red drift algae in 04 and the red tide in 05. They were very similar. And it's the same thing with the blue-green algae. If they're being exported, out as they die, you saw those high concentrations, super high, in the Normandy Canal. I mean, those are concentrations, James, that you would find literally in undiluted sewage, 2,000 micromolar. Okay, I've never seen anything like that. Okay. It was literally like a sewage pond. Okay. So that stuff it's going to be exported out of the estuary because the red tide grows in the coastal waters, not up in the estuary. It doesn't like those low salinity values. So you're absolutely right. It will contribute to red tide biomass. And I think, as you know, we saw super high concentrations of red tide. Compared to historic values in terms of cell counts, millions of cells per liter.
Yeah, thank you for that. I really appreciate it. And that's all the questions I have. Any other questions that we have from the management committee or staff?
Yes, James, our next presenter has graciously said that she's fine starting a few minutes late, so we can have a little bit more discussion here. I had a couple things very quickly. One has to do with that advanced wastewater treatment, 98% removal. As I understand it, there's different levels of what we call AWT. Can you explain, is that the standard, or is that a higher level of AWT than standard? And then the second question has to do with, like, what do we do short of, for those areas where AWT isn't possible or septic to sewer conversion isn't possible? Is there any interim measures like aerobic systems? I think you and I spoke briefly that those are not what was thought as far as being more effective, which is unfortunate. But maintenance or anything else that you would recommend as an interim measure for those areas until we can get to the optimal end result of moving them on to centralized AWT sewering?
Yeah, good questions, Jennifer. The first question, That 98% removal is actually going beyond the AWT standard, which is 3 milligrams per liter of nitrogen and 1 for phosphorus. Okay. So I think, I've been talking with Tom Walker, the engineer who worked with James Barden, who developed the Barden-Foe nitrogen, biological nitrogen removal technology. He was involved in the design of that Cudjoe regional plant. And, Tom Walker lives in Sarasota now. He's not far from you. And you might want to get him sometime as the speaker to talk specifically about the design criteria of these new AWT plants and how they're being able to exceed the standard. I find it really fascinating. And Tom and I were in graduate school together at University of Florida, environmental engineering sciences back in the 70s. He went more the engineering way. I went more the environmental science way. And it was very exciting for me to meet him, of all places, in the Lower Keys. Me working on the problem of nutrient pollution and he working on the other end of the problem, developing the technology to clean it up. So it'd be great to get James Barden also. I know he has come to Fort Myers before and met with one of the professional engineering groups there. And I understand it was a really awesome lecture on these emerging technologies for nitrogen removal. So that's a great lead into your second question, which is, what would I recommend? In terms of on-site systems where you don't, say, have a central collection system to tie into, whether an aerobic treatment unit. And, we have those in the Florida Keys. They're comparable to secondary treatment, meaning they treat the BOD, they aerobically, and you end up with a very nitrified effluent, high nitrate, okay, as opposed to ammonium and high phosphorus. So they don't remove nutrients, they just convert them and remove the BOD. So it's certainly one step better than a septic tank, but it's not removing nutrients, okay? And that nitrate will migrate through the groundwater. So we have this new technology, and there's it's really a technology developed by Siemens Corporation, and there's a company that is currently working with these systems in Florida. They're called On-Site Performance, okay? On-site, SYTE Performance. And I'm working with them right now through FAU Harbor Branch to monitor the installations of these units. And basically, they're like that Cudjoe regional plant on a small local on-site scale. They are engineered for nutrient removal. They have chambers, just like a Barden-Foe system, where you have alternating aerobic, anaerobic chambers to achieve denitrification, biological nitrogen removal, in a system, a concrete box that is basically the size of a septic tank. And believe it or not, these now can be put anywhere in Florida without the need for tying in to a central collection system, meaning we now have the technology to remove nutrients on site pretty much anywhere in the state of Florida. So in these rural areas, on springs, or places that are out of reach of municipal collection systems, to me, this is going to be a game changer, I believe, going forward in Florida. The cost is on the order of $12,000 to $13,000. That's roughly the same price as a conventional septic system. And the ideal scenario is to actually have a utility monitor these systems. You just need the internet, to be able to monitor these systems once they're installed. They're underground. You don't see them on your property. And honestly, I'm very excited that this is really going to help us turn the corner on septic to sewer where collection systems are not available. Okay.
All right, thank you. I don't think we have any other questions, and I don't see any of the raised hands. One final call for questions, and then we'll move on. All right, excellent. Dr. LaPointe, I want to thank you.
There was
go ahead.
just one very quick in the chat. Someone wrote, does that
Okay.
price include the drain field?
Okay, does the cost include the drain field? I guess that's what that question was. I think it does include the drain field. I'll have to check on that, but I believe it does. We just had a groundbreaking in the city of Apopka, and if any of the listeners are interested in more information, go to the city of Apopka website. Of course, Apopka is right on the Wekiva River, and it's one of those spring sheds that's very, very much being impacted by septic tanks. And it was a very exciting groundbreaking there. And so, yeah, I would urge you to go check that out.
The opportunity to ask one more question real fast.
Absolutely, go ahead.
This is Elizabeth Wong with the city of Northport. Is there a document or some study you can share with us on how to use your traces that you mentioned, sucralose and the nitrogen isotope, to prove that septic systems are leaching into waterways?
Well, you could do, we have our published papers that we could, if you could email me at Harbor Branch, I'll be glad to send you the papers that have the method. And we work with Florida International University, the Biscayne Bay campus. They can run sucralose for you if you want to collect them from your surface water or your groundwater. They're, I think, if you do 6 samples or less, they're $100 per sample for the analysis. If you do more than 6, they're $50 each. So I would suggest you do 6 or more, and the price drops by half. And we can, if you reach out to me or just try to go to the FIU site, their website, you might be able to locate that lab. But if you can't, email me and I can help get you set up.
Thank you so much.
You're
I will
welcome.
reach out to you and to the FIU.
Yeah, thank you.
All right, with that, I want to thank you, Dr. LaPointe, for taking the time to present to our group today. Appreciate all the great work you're doing, not only here in Lee County, but throughout the state. And I also want to, thank Lee County for, really focusing on the septic tanks and the impact that that's having on the Caloosahatchee watershed. Really important stuff, and we appreciate all the great work you're doing.
Great. Thank you, James. It's been great. Appreciate the invite. Okay. Bye-bye.